So our work focused on the Theranostic translational imaging using radiolabeled antibodies for PET imaging. And we believe that PET imaging with these radiolabeled antibodies can help in patient stratification regarding the receptor status of people and to follow the biodistribution of these antibodies and to see or to detect off-site accumulation of these antibodies. And our work here presented at the AACR was about lymphoma imaging using the CD19 antibody, and as we see, CD19 has emerged as an excellent target for lymphoma patients with the CAR T-cell therapies or bispecific antibodies or other FC-optimized antibodies, but almost 50% of the people don't respond to this therapy. And with a radio-labeled antibody before therapy start, we aim to select patients which are eligible for this therapy and which might respond rather to the therapy as other patients that do not. We developed a GMP-conform radiolabeling facility at Tübingen, so to be ready for clinical studies. And we have taken much efforts in developing this kappa-labeled antibodies in preclinical mouse models, and we already have had 6 patients measured and imaged for PET imaging in the clinics, and we detected almost many lymphoma sites of these patients in bone marrow, in all lymphoma sites, mediastinal and peritoneal lymphoma sites, and therefore we believe that it's a quite good imaging modality to satisfy those people. And the next steps will be now that we correlate accumulation of this radiolabeled antibody to therapy response. So we try to do first imaging with this CD19 radiolabeled antibodies. Then we offer a CD19 targeted therapy, and then we correlate this PET imaging with the therapy response of this targeted therapy. Definitely. So as we see here, for example, today there are other excellent works doing on this, as Anna Wu's lab, for example. She presented CD4 and CD8 imaging, antibody imaging possibilities to select patients or to see early response to immunotherapies. And I think not only histology is important, we need in vivo receptor status regarding, for example, tumor heterogeneity and receptor heterogeneity, and we think that imaging is therefore very important, and it will develop more and more in the next years with new imaging modalities such as simultaneous PET-MRI technique, which is very sensitive and specific. And so I really believe that it's an upcoming and getting more and more important in oncology and other subjects as well.